up, YouTube? It's your boy, a J, a O, a H, a H, a A, a B, a R, a A, a H, a A, a M. Let's fucking go. Day 2, Bioinformatics. What is bioinformatics? It's like the informatics of the biology of the informatics, okay? Don't ask too many questions. Don't do that shit. Don't do that shit, okay? You don't gotta ask too many questions, you feel me? Shut the fuck up. You understand? You understand? You understand? Shut the fuck up, okay? Shut the fuck up. I'm playing, dog. I'm playing. You can just keep on talking, dog. You can just keep on. You can just keep on doing that, brother or sister or whatever you are. You might be an AI. I don't know. You never know. I don't know. You never know. Let me see what I did on my last session. Cause it's been a while. It's been a fucking while. Copy. Paste. No, fuck that. That's not what I want. Jackie Robinson. Okay. Mm-hmm. Oh, whoa. Okay. You know who's gonna come up? Claude. I'm gonna open Claude. I'm gonna play some music. Is my screen audio muted? Yeah, it's muted. It's muted. Of course, ads. Of course. Focus. Focus. focus, 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 focus. Okay. What's the going? PyCharm, come on. We bringing up the old boys up. Okay, there we go. And motive scanner. So we do have both of those. K-mer analysis and motive scanner. Okay, okay. Okay, what? Oh, bro, I forgot what I was doing. No, fuck. Okay, got it. Can you? It's been a while since I have. I heard a chat. GD Python. What's up? What's up, dog? How you doing, GD Python? How you doing? How's things going? How's geometry dash going? I should change my gaming update on the description. I should change it to Sleeping Dogs every Wednesday, 8:30 p.m. I should do that. Ba-bum, ba-bum, bum, bum, ba-bum. Here's exactly where you are, how you got there. Your pipeline, where you stand. Motive scanner done. Did I do that? I have camera analysis and okay, I downloaded. Okay. Step 4, Auto Finder. Step 4. Well, right now you have okay, Knox motive positions, but the motive position is just an address. It tells you where something regulatory is happening here, but it doesn't tell you what gene is sitting next to it. What? Heard you got a J word? So you could game now, right? Exactly. But instead of playing Lies of P every day, I thought maybe I should bring down the gaming for a bit. I'm gonna play Sleeping Dogs. I don't know if you've seen the gameplay. I'll post the next gameplay stream on the Discord and stuff. But yeah, every Wednesday, 8:30 p.m. for 30 minutes. Sleeping Dogs. I gotta say, it's one of the best games out there, especially the combat. Whoa! Whoa! The combat is fucking rough. But yeah. So I could technically start gaming, but yeah. I should also focus on the studies. So, yep. How about you though? Have you heard of the game Sleeping Dogs? What step 4 means and why it matters right now for what the motive position is just an address. It tells you something in the regulatory is happening. It doesn't tell you what gene is sitting next to it or if finding solves that or if it's an open reading frame. It starts with ATG and ends with a stop codon. By looking at what's downstream of your Knox motive hits, you find the actual gene sequences. Then takes your sequences and blasts them, compare them against rice and known other plant genomes to identify exactly which Knox gene each one is. Your 2 files so far. Yeah, YouTube chat. Apologize, man. This is how it is. I also should turn on the audio. You are not hearing the chat, right? Like the chat audio. I turned it off. I did not turn it off. I turned off the monitor audio coming from the monitor because I was listening to music. And if music is being streamed on YouTube, YouTube is like, no, mister. You can't do that, mister. Mister, I appreciate you for using YouTube, but you can't do that. No, sir. Arda A3, I'm guessing that's GD Python. Did you finish your lab journal? I did finish. Oh, that was fucking a nightmare. But I did finish it, and I'm actually I'm 100% sure that there should be some place that I should do repetition. So they're gonna correct it, and they're gonna tell me, yo, this part ain't it. You gotta redo it. So that's gonna come up. But yeah, I got 5 exams coming up. I made a new wallpaper. That's my wallpaper, and look at that. Those are dates of the exam. Oh, that's cool. 5 exams. So, what was I doing? I was working on my Working on my project. What? You got project? Yeah. That's what I'm saying. Yo, what happened to Claude? Like, the fuck is wrong with this? Okay, now it's working. Okay. So I have to ORF finding. Right now you have NOS positions, but your motive position is just addresses. It tells you something regulatory is happening here. Okay. So these two are right now two Python programs. And how do I pursue? Next, also I'm wondering, is the next step related to the program, like the outcome from this program? I meant, should I connect these programs for the next step? The professor is basically saying seminar, analyze bio-LSTM, understand the paper and technique. Okay, you know what? On the other side, I should open the bio-LSTM. Where is it? Bamboo? No, not bamboo. Untitled folder. No, not this, not this. Yeah. Oh, there we go. Great question. Yes, the next step uses the output from the motive scanner directly. Let me explain how they connect, how the programs relate, camera analysis.py. GD Python, what was I thinking? Are you still on the chat? Do you have any subreddit recommendations? Because next up, it should be more. And I don't have any subreddits. I mean, I could redo r/memes, you know, that's like infinite content, but come on, I gotta find some niche subreddits. Do you know any? You don't need to re-enter the motive scanner. The Aura Finder reads the saved output and uses it as a starting point. You don't need to merge the programs into one. Each program in your GCNR project is one job cleanly. Okay. So think of it like the factory pipeline. Each program is independent but feeds the next one through the save files. What Aura Finder will specifically do: reads the motive scanner tab text, reads will filter. Wait, your motive scan results dot text has 3 Nox hits. Running or finding on all of them would take hours, so we'll filter first. Only look at the Nox hits that also have T. Wait, could you explain again the last part? Because I need more Random TCG, okay. Look 200 bases upstream. Nothing special, just random sequences. Probably not a real gene, real Knox gene. Look 200 bases upstream. TATTA is here. There, the promoter is present. Very likely for a real gene. Now you check the 200 base upstream of each of these TTA. Why is it like that with the 200 bases upstream? Mm-hmm. Eight minutes. Like, how is this looking biologically? I'll ask ChatGPT on this, right? Or let's bring in Gemini. No, let's bring ChatGPT. Could you explain this? DNA is directional. Okay, okay. What do you mean 90%? GD Python, I'm guessing you don't have too much recommendation. It's understandable. I literally am out of messages. Nope, that does not work. What you're describing is a basic promoter check. Biologically, it makes sense, but it's not a strict rule. Here's the actual meaning. Genes are read. Oh, it's over. Mm-hmm. Genes are read starting at a transcription start site. You know what? Can I use Sonnet 4.5? I gotta reply. Fuck. Nope. Nope. Genes are read starting at the transcription state. Come on. That's not Gemini. 15 minutes. going to be recognized by TBP, Tata binding protein. This helps recruit RNA polymerase 2, which actually starts transcription. Oh, come on, I pressed enter, bro. Come on. Data box is recruits machinery, gene gets transcribed. Okay, are we talking here? Are we talking when the M to make a certain protein or enzyme or is it inside the DNA where this is what starts with the production for mRNA. I'm still new to this subject. Since you run the code and have results, here's what they mean and how to interpret them. What these results represent. By running this specific script, you filter down. The anatomy you confirm hits. A candidate is only confirmed if it meets the 3 physical criteria. Switch the promoter that tells the cell start reading here. They're mixing two states together, let's separate them. So where everything happens, all this happening in the DNA inside the nucleus, nothing starts in the mRNA. What TATA box actually does, TATA box is the part where DNA, not RNA. Its job protein called TBA binds to the DNA region. It attracts other proteins, forms a transcription complex. This complex brings the RNA polymerase 2. What happens next? This is the key step. Once everything is assembled, DNA, gene, RNA polymerase reads it, mRNA is made. So yes, the output is mRNA. mRNA later becomes the protein, full flow, very important. Okay, so where inside here would that 200 base pair gap be? What? There's no. Where is the Gemini app though? Okay. Is that even real though? I don't know. That's the problem. I don't really know. Okay. Fuck, how do you? This is upstream of the gene that includes regulatory. If I understand, the starting step would be regulatory part, or the regulatory sequence. Now I'm wondering, is this also the thing that decides how long each cells replicate, etc.? Mata. Can you get one more screen? Please. I think the phone sharing part, that's where it's causing an issue. Okay, so okay, I can live with that. Do use local blast, okay. Would you like to see a scripture translate these scenic candidates and promising? Wait. try it now. Top candidates are around ORF within ORF links. Scientific context, okay. Cell replication, it's controlled by cell cycle genes, signaling pathways, environmental cues, examples of key regulators, cyclins, CDKs. These are different genes entirely, but do these genes, okay. Could you explain what the synonyms should control like these genes gene I care about, ORF. Does this look like a real protein coding gene? Not replication related. The core misunderstanding. These genes should control replication of the or suppression of the other genes. Half correct but wrong mechanism. Two completely different processes. DNA replication copying the gene. DNA replication copying the gene. Turning genes on or off, but what does that? They bind DNA and decide activate this gene or suppress this gene. Regulatory DNA, your base pair region. This is where the proteins bind. Okay, this is where the proteins that make the mRNA bind, okay. Strength of expression. Epigenetic, this is what you were hinting at. This is the closest at what you described. Instead of removing genes, cells chemically modify DNA or histones, make regions more or less accessible. So histones is like a hard disk. Ten minutes. You cannot run a blast search in vacuum. You need to check to ensure you're where to get them, what to search, look for. It's not built-in DNA timer, it's mostly signals, both external and internal. The cell interprets. Two types of signals control this. An external signal, very important in plants. These come from outside the cell, plant hormones, signals from neighboring cells, environmental inputs. Examples: auxins promotes growth at tip, cytokinin promotes cell division, gibberellins elongation, so at the bottom tip. So at the bamboo tip, division on, further away, lower signals, division off, okay. Internal signals, cell state, inside the cell, protein levels, energy state, DNA damage, sex, developmental programs. Protein levels, energy status, DNA damage checks, developmental programs. Example, cyclic cell levels drop, CDK inactive cell cycle stops. Cyclin. Is there a timer? Not really a clock like after 10 divisions stop. Instead, it's more like continuous sensing decision making. Cell constantly asks, do I have growth signal? Do I have nutrients? Am I in the right position? Position matters, critical for plants. And something like bamboo cells at the tip receive growth signals, strong growth signals. As cells move away, they lose access to those signals. Location, signal exposure, gene activity, cell behavior. Where DNA fits into this. DNA does not act as a timer, but it contains genes for receptors. So DNA provides machinery, not the timing. Putting it all together, external signals, hormones, neighbors, internal state, proteins, enzyme, energy. Activate, inhibit transcription factors, turn genes on or off cycles, cell division or differentiates. Why is this important for your thinking? Okay, but how is... But I'm kinda confused on how is those external factors actually changing how the data from the DNA is being... Like, how is the external factors communicate? I thought the nucleus has a cell wall and it's for things to go in. So how is that signaling work? If option A, blast and nucleotides to nucleotides, how it works compares your DNA directly to DNA of known plants. Very fast changes. DNA changes faster than proteins. If your GC in our organism is distantly related to the reference, blast might miss it. Interpreting the side results when you run blast, you'll get a check. And also only Shut up. ChatGPT just keeps on glazing. The answer is signals don't go directly to DNA. They go through their relay system. First correction: nucleus is not sealed off. Nucleus does not have a solid wall. It has a nuclear membrane with pores. These pores allow proteins signaling molecules to move in and out in a controlled way. How the signal travels from outside. Signal arrives outside the cell. Hormone like oxygen inside cell. Signal molecule detects it using receptors on the cell membrane. Special proteins called receptors bind the signal. Signal bind receptors. Receptors activated. The signals gets passed into inside signal cascade. This is like a chain reaction. Receptor, protein A, protein. Each step activates the next protein, amplifies the signals. Activation of the transcription factors. Eventually, this cascade activates a protein that can affect genes. Transcription factor examples: transcription factor. Transcription factor enters nucleus because of the nuclear pores. Transcription factors enters nucleus. Did I just read the same thing? Yep, I did. I did. Since okay, 15 minutes. Yo, Arda833. Okay, it's GD Python. By the way, never left the stream. What's going on? Interesting, you asked me. So, how do I explain this? I'm still thinking. How plants work, how they replicate themselves. So I was trying to understand what they were doing. Right now, I'm using an example of bamboo. 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 Let me see bamboo. There we go. There we don't go. No, they did not show me a bamboo picture. Yup. I was trying to understand how this thing replicates itself, how it makes itself. So, So what did this program did right now? It tried to find positions where inside the DNA it could result in a gene. Did you get it? So for 19,334 Knox hits, 62 confirmed gene candidates. So right now I don't know exactly what the Knox hits are. I believe they are positions in the DNA, and from the 19,000 positions in the DNA, there were 62 confirmed. That is just for this part, okay? So there were 62 confirmed gene candidates. I don't know what type of genes they're resulting, but yeah. So my next step would be compare these gene candidates or these gene positions with other plants and see how similar these fuckers are. Why am I doing this? It's all gonna make sense. I hope so. But hey, GD Python, I appreciate you for hanging on. Means a lot. What was I thinking? I got 15 minutes. I should run blast. What is the plan I should be going for? So, transcription factors enters nucleus. Now it reaches the part you're working with, binds upstream region, your 200 base pair zone. This changes where RNA polymerase binds, whether the gene is on or off. Wait. Wait. So you're telling me these transcription factors are just what? So these transcription factors kind of hide. So these transcription factors kind of hide. Hide the said. What's the name? What's the name? What's the name? Fuck. Regulatory. Hide or express. More of the regulatory gene, which results in production of the which result in the production of the mRNA for that. Is that what it is? I don't know. Around the world and around the world and around the world and around the world. It's 10 and I should be sleeping. I have to read the German, this thing. Frittatensuppe, Leberknödelsuppe, Kürbis-Cremesuppe, Fleisch- oder Speckbrot, Schinkenkäse-Toast, Schinkenkäse-Toast, Actionschlägers Classical von Mittwoch bis Samstag, gebratene Blunz, saure Schweinsleber, Cordon Bleu, Leberschädel, Rauber, Räuberteller, Kinderwiener, ein Haschig-Knödel, Süber gemischte Haschig-Knödel, gebackene Barkändelsalat, veganes Chili sin Carne mit Reis, Forellenfilet. Oh yeah, I remember that one. Bauernkarpfen, Sacher, Orte, Topfstrudel, Povidel, Taschel, Eispalatschinke, Sirbenschnaps, Obstler Nussschnaps, Most pur oder gespritzt. Most pur. The fuck is that? Most Radler mit Almdudler. Did I just read Dudler? Dudler? Oh, we still got more. We still got more, baby. Baby. You're basically describing the real system now. Okay. Now I get to decide, should I continue or should I not? This did not take too much time, right? Yup. I'ma end the stream because... Wait, we got a Reddit page? What the fuck is it? Yep, I'm gonna end the stream, gang. It ain't much, but it's honest effort. I'm gonna end the YouTube stream. I'm gonna continue with German. Peace out.